Hello student, very good morning to all of you. How are you? I hope you all are happy, healthy and fine at home. Do you remember what were we discussing yesterday? I think we were discussing about the longifulin and what will be the structure of longifulin, and first of all we are discussing about the retrosynthetic analysis of longifulin. And as you know that longifulin, which is a natural product, and as we know that natural products are generally obtained from the various parts of the plants, and they found to be very useful as per as medicinal point of view is concerned, and that's why we are studying this type of the selective representative natural product. And when we are extracting this such type of the natural product from the nature, from the plants, so it will be what we can call it as is not much we cannot extract in that quantity, whatever quantity it required as a medicinal point of view, and for that the same structure the scientists trying to synthesize in the laboratory, starting from the known as well as easily available starting material, and we can prepare it on a larger scale. That is the main purpose of this synthesis, and therefore we are trying to synthesize. Let us see first one is a retrosynthetic analysis. Retrosynthetic analysis means what? As we know that there are the various different terms used during retrosynthetic analysis. So let us see once again, that is a target molecule. What will be the structure of longifulin? As we know that the structure of longifulin will be this one. Let us see, this is a structure of longifulin. See carefully here, you can clearly see this is a structure of longifulin where there is CH2 double bond and one methyl group over here. So there are, this is our target molecule, and target molecule means what? Whose synthesis being planned is called as a target molecule. If you number the carbon atom, you can find out the total number of carbon atom over here. So 1, 2, 3, 4. So here let me make the correction over here. Here is a directly joined over here. 1, 2, 3, 4, 5. So this carbon atom, let me drop properly over here. This is a carbon atom 7-member ring because it is a tricyclic compound. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, then 12, 13, 14 and 15. There are 15 total number of carbon atom. 15s are present there. As we know that there are the 3 cycle, means it is a tricyclic and it is a hydrocarbon. Hydrocarbon means what? The organic compound which is made by carbon and hydrogen is known as a hydrocarbon. And then how many chiral centers are present here? This compound is optically active in nature and it has 4 chiral center. Chiral center carbon number 1 and carbon number 2 are chiral center. Similarly, carbon number 7 and carbon number 10 are the chiral center, and each chiral center is not dependent on each other, they are separately, and therefore the stereochemistry plays an important role here. And this is our target molecule Tm, and now we are doing retrosynthetic analysis. As we know that retrosynthetic analysis means what? The process of breaking down the target molecule into convenient starting material is known as retrosynthetic analysis. And now we know that this is a disconnection arrow. It is a special arrow. The meaning of this arrow, this target molecule could be made from this, and that is the general meaning of this arrow. And now after here you can write FGI, functional group interconversion, means one of the functional group converted into as another functional group, so that disconnection becomes as a possible FGI. Then here you can use as a this type of the then Wittig approach we can use here. Wittig approach, similarly approach, as well as we can use one another, that is pinacolon rearrangement, and by using as these two approaches you will end up with this compound is like this. So here you are going to get the compound is this one. Let's see, this is a compound which I have drawn over here. This is a 7-membered ring, 1, 2, 3, 4, 5, 6 and 7. 1, 2, 3, 4, 5, 6 and 7-membered ring. Here methyl group as it is. Here what is happening? Here only one methyl group over here. Here we are introducing as a carbonyl group. Similarly, this carbonyl group is present over here, and now this compound we can call it, this is a compound number 23 which is given in the book, and this is a target molecule, suppose as a 23. So now this 23 and this compound they called as a compound number 23 first, you can call it as, and this compound number 23 first. Then we can do the retrosynthetic analysis once again. Functional group interconversion. Functional group interconversion means one of the functional group is converted into another functional group, so that disconnection becomes as a possible. And therefore, so what here we can use as one of the approach, like as a pina. Here this bond is broken over here, so you may get that tricyclic is converted into as a bicyclic, so you get the structure over here, this type of the structure. You may here may find this is a structure where C double bond O, this methyl group remains as it is. What is happening when removing from here, the double bond comes over here, one methyl group over here, and this is a carbonyl group which remains as it is. So this is the compound which we got over here. This is alpha beta unsaturated position. You just unfold it, and this we can call it as a structure number 23rd second. And if you unfold this this is a 6-membered ring and this is a 7-membered ring. There are the 2 rings, and that 2 ring, if you unfold systematically, then I am showing over here this 3 arrow means this structure I am writing over here is, let's see, this is a 6-membered ring. I am writing as it is here. This is a 6-membered ring. This 6-membered ring I am written like this. Here is a C double bond O. Now this junction, and this is this part, right? This part, there will be a one methyl group. That methyl group I am showing like this stereochemistry over here, and this is a bond, this bond is there. So let's see, suppose this is a, now if you remember as a numbering system over here, 1 2 3 4 5 6 7, then 8 9 10 11, and this is a 12, right? Now this is a 12 actually, and this one is a 11 number, right? 11 number. Now this 5 carbonyl group, so we can this unfolded, you can show this ring as it is over here. This is a ring which is a 7-membered ring. 1 2 3 4 5 6 7-membered ring. Now look at this ring I have drawn like this. The methyl group which is there, this is a, what we can call it as 11 number. This one is a 2 here. This will be 3, then 4. Now 5 means here there is a 5, and then 6, there is a double bond, 6 here. Now 7, this one, and here this will be the 1 2 3 4 5 6 and 7, right? And now this one 8, then 9, this is a 10 and 11. So what we did, we just unfolded and we got this type of the structure over here. And this, if you see clearly, there is a enone system or the alpha beta unsaturated compound, and this structure, this 23, this is also as a 23 second structure, right? This is a 23 second structure. And after this one 6-membered ring, this again you can carry out the FGI functional group interconversion, and then from second you will get as a structure number 23, third structure. Now here by using some special approaches, this 7-membered ring we can convert it into as a 6-membered ring. So let's see over here. So you are going to get that one of this type of the compound, this remains as it is, C double bond O. This methyl group also remains as it is, but here are the changes we made. Now this double bond now shifted over here, and at this position there will be a, what we can call it as here, hydroxy group OH, and here is a CH CS3, and here we can attach as a X group. So by doing some rearrangement, maybe as a Verlep and drop rearrangement approach we can use over here. So 7-membered ring can be converted into as a 6-membered ring. This type of the target molecule you may get over here, and this is known as a structure number 23 third, right? These are Roman letters I have selected over here, and that if you see carefully, you will find there is a hydroxy group, here is X, and they are present at vicinal position. And now this third structure we can convert by doing the retrosynthetic analysis. Again you can use as a functional group interconversion. So here you can bring as a hydroxy group, we can remove this one. Instead of these two functional group, we can bring there is a hydrogen atom, so that you will get another structure, this one. So this is a structure where double bond here, here also has a double bond, and then here you will get as a this CS3 group, right? And now other part of the compound, it is remains as it is. So like this. So this is a structure number, we can call it as a structure number 23(4). This is a structure number. What did we do? We removed that hydroxyl group from here, we bring as a double bond. That's the changes we made over here. And after that we can do once again as a functional group interconversion. See here, there is a double bond, we can brought there carbonyl group, and which become as a this type of a compound here, and we will get this one. Here is a methyl group and this carbonyl group, it is as it is, this double bond, and here oxygen atom. This is alpha beta unsaturated compound. This type of the compound we can prepare by using as a aniline approach or the Michael addition type of the reaction, and this is a compound which we can call it as a, this is a structure number 23(5). So this compound is readily available in the market, and therefore starting from this we can achieve, we can obtain this target molecule. So this is the retrosynthetic analysis by using as a various approaches we arrived up to this, and that is known as a retrosynthetic analysis. Retrosynthetic analysis is nothing but, what is that? This is a scheme 23, scheme number is represented over here. Retrosynthesis of longifulin corresponding to the synthesis in scheme number 24. Synthesis we are going to learn. There are 9 such syntheses. So let's see some introduction regarding this. The first synthesis of longifulin was described in detail by E.J. Corey and co-workers in 1964. Scheme 13.23 represents a retrosynthetic analysis corresponding to this route. A key disconnection is made on going from 23 first to 23 second. Look at here, from here to here, a key disconnection is made on going from 23 first to 23 second or first to second. This transformation simplifies the tricyclic to a bicyclic ketone. This is a tricyclic when we got second compound, which becomes as a bicyclic. Bicyclic is a simpler than the tricyclic, and we can easily obtain that bicyclic skeleton. And for this disconnection to correspond to a reasonable synthetic step, the functionality in the intermediate to be cyclized must be engender mutual reactivity between C7 and C10. C7 and C10 we have broken from here. So this bond is broken and we will get as this one. And this is achieved in diketone 23. Diketone, there are the two ketonic groups are there. Diketone 23, diketone 23. Second, because an enolate generated by deprotonation at C10 can undergo an intramolecular Michael addition to 7. C7 Michael addition, we know that Michael addition are 1,4 addition. It is similar to as a aldol condensation reaction. Michael addition is the nucleophile approaches at carbon-carbon double bond side, and that type of the reaction are known as intramolecular Michael addition to C7, and we will get this one. The stereochemistry requires that the ring junction be cis, and if cis, just like as a decalin which is a cis in nature, means all the ring carbon they are lying in the same plane, and that is known as a cis. Retrosynthetic step 23 second converted into as a 23 third is attractive because it suggests a decalin derivative as a key intermediate. Methods for preparing this type of structure are well developed since they are useful intermediate in the synthesis of other terpenes as well as steroids. Can a chemical reaction be recognized that would permit 23rd to 23rd second to proceed in the synthetic sense? The hydroxyl to carbonyl transformation with migration correspond to the pinacol rearrangement, and you might be knowing as a pinacol rearrangement, and by doing that we will get as a ketone, and that's why this type of the rearrangement is helpful, useful for the synthesis of longifulin. And the retrosynthetic transformation 23rd second changes to 23rd third correspond to a workable synthetic step if the group X in 23 third is a living group that could promote the rearrangement reaction. The other transformation in the retrosynthetic plan 23rd third to 23rd fourth to 23rd 5 are straightforward. There is no any confusion. It is very straightforward in concept and lead to identification of 23,5 as a potential starting material. This is a very potent or the potential starting material used for the synthesis of longifulin. So here if you see carefully, if you take this is an starting material which is readily available, if you do here, if you introduce this type of the carbon-carbon double bond over here, what you can do? So first of all, you have to protect this carbonyl group. If you compare the reactivity between these two carbonyl group. One is a isolated carbonyl group, whereas another one is a conjugated carbonyl group, means alpha beta unsaturated carbonyl group. If you compare the reactivity between these two carbonyl group, you will find this isolated carbonyl group will be more reactive as compared to the alpha beta unsaturated carbonyl group toward, and therefore it is required to protect this carbonyl group and then carry out the reaction on this carbonyl by using as a modified Wittig reagent. We can get this type of the compound, and once you get this type of the compound, then you can use as osmium tetroxide, where we know that osmium tetroxide is well known for getting cis hydroxylation. So you will get here cis hydroxylation, and once that hydroxylation, one is a tertiary hydroxyl group you will get, and second one is a what we can call it as a secondary alcoholic group you will get. And now one of them we can protect, hydroxyl group can be protected by different protecting groups, but here selectively we can use tosyl chloride in presence of the pyridine, and one of the hydroxyl group preferentially selectively get protected among these two, the less hindered hydroxyl group, which is a secondary hydroxyl group, which will get protected very easily, and then you can carry out the reaction like as a rearrangement reaction, pinacolon rearrangement. You will be end up with this type of the compound, and then you can get some using as a Wittig approach pinacolon rearrangement. We will get that our compound, that is as we might be knowing as a intramolecular Michael addition reaction, then pinacolon rearrangement reaction, how it can be carried out. For these are maybe as a compound 23,5 is known as the Wieland-Micheler ketone and can be obtained by Robinson annulation of 2-methyl cyclohexanone 1,3-dione, means the name of this compound, the starting material also is known as the Wieland-Micheler ketone and can be obtained by Robinson annulation of 2-methyl cyclohexane 1,3-dione. So in that way we can get this, and this is a further. Now we will we will start the synthesis of the compound, which will be starting from this synthesis was carried out as shown in scheme 23.24. Now we are going to synthesize this. This is our, let us see the first synthesis, how we can synthesize that longifulin here. So that's the thing which we are going to discuss over here. And now let's start the synthesis of that longifulin. Let's see here. the first synthesis, that is a 13.24 scheme number, and where the first synthesis we are starting over here. So let's see the starting material will be this one, as we know that carbonyl group, one of the methyl group, double bond over here, and this is a compound. This is a compound. When this compound further treated with one reagent we are going to use over here, this one hydroxyl group, second will be PH3 P double bond CH CH3, that is a second reagent which is a modified Wittig reagent, and the third reagent we are going to use osmium tetroxide OSO4, and the fourth reagent which we are going to add, that is a TSCl, tosyl chloride, paratoluene, sulfonic chloride, that is in short represented as TSCl. And now by doing this, what do you get? If you see, these are the compound carefully. When you add as a first reagent, what do you get? There are two carbonyl groups are there, two competitive groups are there. Which one will shows as a greater reactivity toward this? So that diol is a reagent used to protect that isolated carbonyl group. It get reacted easily, and you will get the product like this. And now here is a methyl group, double bond over here, and this is a double bond which remains as. After that, when you add second reagent here, which is PH3 P double bond CH3, which is a modified Wittig reagent, what is going to happen? It is going to react with this, and you will get this type of the product. This one remains as it is, methyl group as it is, this double bond as it is, and here you are going to get, here is a CS3, right? After doing this, then osmium tetroxide OSO4, and as we know that osmium tetroxide is well known for dry cis hydroxylation, and you will get the product over here, this one. This you can write as it is OO, like this, methyl group, and here CS3 OH, and here OH, and after finally you are going to add TSCl, generally in presence of the pyridine, the reaction. Now there are again two options. OH, it is a secondary, and this is a quaternary. Which one is going to react? This is a tertiary, this is a secondary. Secondary get protected, and finally you are going to get the product over here, this one. This is the product which you are going to get over here. This double bond remains as it is, hydroxy group OTS over here, and this methyl group. This will be the product which we are going to get after adding this reagent. Understood this one? And after adding this, we will get hydroxyl group, then H plus acid workup, and you will end up with the another. I think this is a thing which we have discussed over here. Remaining we will see in the next lecture, tomorrow's lecture. Let us stop today here. Have a nice day. Thank you.